Hello students, erythropoiesis, production of red blood cells. Let us learn different stages of erythropoiesis in this video. Erythropoiesis actually is a process of origin, development and maturation of red blood cells. It is different from hemopoiesis or hematopoiesis. Hemopoiesis or hematopoiesis is a process of origin, development and maturation of all blood cells, whether they are white blood cells, platelets or red blood cells. Before starting the process of erythropoiesis, let us study the sites of erythropoiesis, where exactly this process takes place. In fetal life, during first 2 months, RBCs are produced by mesenchyme cells of yolk sac, and this stage is called as mesoblastic stage. From the 3rd month onwards, the liver takes over the job of producing RBCs along with other organs like spleen and lymphoid organs, and this stage is called as hepatic stage. In the last 3 months of intrauterine life, RBCs are produced by red bone marrow and liver, and this stage is called as myeloid stage. While in newborn babies, children and adults up to the age of 20 years, red blood cells are produced by red bone marrow of all bones, long bones as well as flat bones. And after the age of 20 years, the red blood cells are produced by membranous bones like vertebrae, skull bones, iliac bones, sternum, ribs, scapula and at the ends of long bones. Let us start with the process of erythropoiesis. Blood cells begin their life in the bone marrow with a single type of cell called pluripotent hemopoietic stem cells, from which all the cells in the circulation are eventually derived. Pluripotent hemopoietic stem cells undergo divisions and give rise to 2 types of cells. Number 1, lymphoid stem cells, which eventually give rise to T lymphocytes and B lymphocytes. And second, myeloid stem cells, which give rise to 3 types of cells: erythrocytes, about which we are going to study in this video, then granulocytes like neutrophils, eosinophils and basophils, and blood platelets. The journey of myeloid stem cells to mature erythrocytes include following stages. These are the different stages. Number 1, proerythroblast. Number 2, early normoblast. Number 3, intermediate normoblast. Number 4, late normoblast. Number 5, reticulocyte, and finally matured erythrocytes are formed. Myeloid stem cells undergo cell division and give rise to first stage called as proerythroblast or megaloblast. This step of erythropoiesis is stimulated by a protein hormone called erythropoietin, which is mainly secreted by kidney in response to hypoxia. It is a condition when oxygen level is less in the tissue. This proerythroblast is the largest cell among all stages and has diameter equal to 20 microns. Its nucleus is also very large with 2 or more nuclei and reticular network. There is no hemoglobin in these cells, but synthesis of hemoglobin begins at this stage. Cytoplasm is basophilic in nature. It further multiplies several times and forms next stage of erythropoiesis. Second stage is early normoblast. Cell of this stage is smaller than proerythroblast with diameter equal to 15 microns. You can see this is the diameter of the cell. It is 15 microns. In this stage, nucleoli disappear and condensation of chromatin network occurs. You can see here, this is the nucleus and the condensation of chromatin network takes place further, and this condensed network becomes dense. Next, cytoplasm is basophilic, and that's why it is also called as basophilic erythroblast. Here also, cell shows active mitosis and form next stage called as intermediate normoblast. Intermediate normoblast is also called as polychromatophil erythroblast. Size of cell further decrease and is smaller than early normoblast with diameter ranges from 10 to 12 microns. Nucleus is still there. You can see here nucleus is present, okay, and chromatin network further condenses. Also, cytoplasm is basophilic in nature, and this is the first stage when hemoglobin starts appearing. Thus, cytoplasm can be stained with both acidic as well as basic stains. Next is late normoblast, also called as orthochromic erythroblast. Here the size of cell further decreases and diameter ranges from 8 to 9 microns. Now check the nucleus. Nucleus is very small. It looks like an ink spot nucleus with much condensed chromatin network. Now check the hemoglobin. The quantity of hemoglobin increases and cytoplasm is now acidophilic. And in the final stage of late normoblast, nucleus disintegrates and it disappears by the process called as pycnosis. Okay, so this nucleus disappears in the late stage or in the final stage of late normoblast. After late normoblast, next stage of erythropoiesis is reticulocyte, also called as immature red blood cells. Reticulocytes develop and mature in bone marrow and then circulate in bloodstream for about a day. They are slightly larger than mature red blood cells, and cytoplasm of reticulocyte contains reticular means mesh-like network of ribosomal RNA. You can see here, this is the reticular network of ribosomal RNA which can be stained with supravital stain. And due to the presence of this reticular network, it is also called as reticulocyte. Finally, reticulocyte develops into matured erythrocyte in bloodstream. Here, reticulate network disappears. Cell becomes matured red blood cells and shape becomes biconcave. Now check the size. Size is equal to 7.2 microns in diameter. It is with hemoglobin. Hemoglobin is present in erythrocyte, but it has no nucleus. It is without nucleus. It requires 7 days for development and maturation of red blood cells from proerythroblast. And out of this, 5 days up to reticulocyte. From proerythroblast to reticulocyte, it takes 5 days, and from reticulocyte to red blood cell, it takes 2 more days. Clear? This is the summary of whole process of erythropoiesis, starting from myeloid stem cells in bone marrow to erythrocyte in bloodstream. Myeloid stem cells first form proerythroblast in the presence of hormone erythropoietin secreted by kidney in response to hypoxia. In proerythroblast, synthesis of hemoglobin begins. Nucleoli disappears at early normoblast stage. Hemoglobin appears for the first time at intermediate normoblast stage. In late normoblast, nucleus disappears. In reticulocyte, reticulate network of ribosomal RNA is present and cell enters in bloodstream where it is converted into matured erythrocyte. Matured erythrocyte does not contain reticulate network of ribosomal RNA and cell shape is biconcave with hemoglobin present in it. So, this is the whole process of erythropoiesis.